Welcome back to the deep dive. Today we are putting away the simple Mendelian rulebook. We're exploring the parts of the human genome that, well, they just flat out refuse to follow the rules of inheritance we usually talk about.
That's exactly right. When most people think genetics, they think, Punnett squares, a dominant gene, a recessive one,
Yeah.
clear 50-50 odds. Simple.
Oh, yeah.
But the disorders we're diving into today, they are much more sophisticated exceptions to those rules.
So our mission today is to really understand four genetic mechanisms that create some pretty baffling and frankly critical disease patterns. Things like anticipation, where a disease actually gets worse with each generation, or parent of origin effects, where it really matters if you got the gene from mom or dad, and even diseases unique to the mitochondria, the cell's powerhouses.
And understanding these, let's call them non-classic rules, is absolutely paramount. Why? Because they often affect the tissues that need the most energy, the brain, the heart, muscles. So these mechanisms, they aren't just biological quirks. They're really the keys to figuring out why certain developmental disorders, certain neurodegenerative conditions show up the way they do.
Okay, let's unpack this then. We're focusing on four main categories today. Those tricky trinucleotide-repeat disorders, then mitochondrial mutations, genomic imprinting, and finally the complexities of something called gonadal mosaicism.
Sound good.
Let's start with the one that causes some really perplexing neurodegeneration, the repeats. What's going on there?
Right, trinucleotide-repeat expansions. These disorders are basically driven by DNA instability, specifically stretches of 3 DNA letters repeating over and over, usually G's and C's.
Okay.
And this instability means the sequence can actually get longer, it expands as it's passed down through generations. We know of, I think, around 40 diseases linked to this now. And they are overwhelmingly conditions that affect the nervous system.
What I find fascinating here is how the inheritance pattern seems, well, weird. If these are technically dominant or recessive, why would the sex of the parent passing it on matter? Isn't DNA just DNA?
That's the first really surprising principle here. The instability is often sex-dependent. The chance of those repeats jumping from, say, 40 copies to hundreds is strongly influenced by which parent transmits it. So for Fragile X syndrome, the big expansion, the one that causes the full disease, that happens almost only when the mother passes it on, during egg formation oogenesis.
Right.
But for Huntington's disease, the expansion is much more likely during the father's sperm production, spermatogenesis.
Okay, that immediately explains anticipation, then the disease getting worse or starting earlier in later generations. But you mentioned another principle, the way the repeats actually cause damage. You said that gets really interesting.
Oh, yeah, the molecular pathology.
Yeah.
Because it's not just one way. There are 3 really distinct ways these repeats can be toxic to cells.
3 ways.
The maybe simplest route is a loss of function. If the repeats are piling up in a part of the gene that doesn't code for protein, they can trigger something called hypermethylation. It's like putting a permanent chemical lock on the gene, shutting it down completely. No protein gets made. That loss of protein is the core issue in Fragile X syndrome, FXS.
Okay, loss of function. Makes sense. What else?
Then there's the classic toxic gain of function,
Right.
but this time in the protein itself. Think Huntington's again. The repeats are in the coding part of the gene.
Right.
So the cell makes this messenger RNA, translates it,
Right.
and the resulting protein has this long, abnormal, sticky tail of glutamine amino acids, a polyglutamine tract. These proteins slump together, gum up the works in the nucleus, interfere with other genes, basically poison the cell.
Nasty stuff. Okay, that's 2. What's the third?
And the third one is, I think, the biggest conceptual leap discovered more recently. It's a toxic gain of function, but it's mediated by the RNA itself.
Wait, the RNA, not the protein.
Exactly. In this case, the gene gets transcribed into messenger RNA. Maybe the repeats are in a non-coding bit, so the protein itself might be okay, or maybe you're in that pre-mutation range we'll talk about.
Okay.
But the messenger RNA molecule, with all those extra repeats in it, is toxic on its own. It acts like a sponge or a trap, soaking up essential RNA binding proteins in the cell. So these vital proteins get stuck to the faulty RNA and can't do their normal jobs elsewhere in the cell.
Wow. That is a huge shift. The damage isn't from a bad protein. It's from the intermediate message itself. That's really something.
It really is. That understanding is key. Let's use Fragile X syndrome, FXS, as the main example here. It's the leading inherited cause of intellectual disability in males.
Okay.
It involves the FMR1 gene, which has CGG repeats. Normally, people have, say, 6 to maybe 55 of these CGG repeats. Totally fine.
Right. But then you get the carriers.
Exactly. If you're a carrier female, or what's called a normal transmitting male, you're in the premutation range, roughly 55 to 200 repeats. Still usually phenotypically normal for FXS itself.
But the problems start when you cross that threshold.
Yes. When it expands beyond 200 repeats, sometimes up to 4,000, that's the full mutation. That's where you get that gene silencing we talked about,
Right.
the loss of function. You lose the FMRP protein, and that's what causes Fragile X syndrome.
And the consequences of losing FMRP.
Well, FMRP is critical for regulating protein synthesis right at the synapse, the connection between neurons. Losing it fundamentally messes up synaptic plasticity, the brain's ability to adapt, which is essential for learning and memory.
So profound intellectual disability.
Marked intellectual disability, yes. And often characteristic physical features, like a long face, large ears, and in postpubertal males, enlarged tests, or macroorchidism.
But what about that premutation range, the 55 to 200 repeats? You said they were usually okay for FXS itself, but there's a paradox.
Yes, the premutation paradox. This is where that toxic RNA mechanism comes roaring back, those 55, 200 repeats. They aren't enough to fully silence the gene and cause FXS,
Right.
but they are enough to produce that toxic FMR1 messenger RNA.
Ah, the RNA sponge effect.
Precisely. And that drives completely different problems. In older males, it can cause Fragile X-associated tremor ataxia syndrome, or FX-TANS, a progressive neurodegenerative disorder.
Wow.
And in about 20% of female carriers, it causes Fragile X-associated primary ovarian insufficiency, basically premature menopause. Different diseases, same gene, different mechanism linked to repeat number.
Okay, that really clarifies the different ways repeats cause trouble. So sex-dependent expansion shows which parent matters for instability. Like, let's shift. What about things happening outside the nucleus? You mentioned mitochondria.
Right, let's leave the nucleus entirely.
Yeah.
Your mitochondria, the power plants of your cells, have their own small circle of DNA, mtDNA. Only 37 genes, but absolutely vital for making energy.
The key inheritance rule here is...
Strict maternal inheritance. The mother's egg cell contributes almost all the mitochondria to the fertilized zygote. The sperm contributes practically none that survive.
Okay, so mothers pass their mitochondrial DNA to all their children, boys and girls.
They're right.
But only daughters can pass that mtDNA on to the next generation.
Exactly. Sons inherit it, but they don't transmit it.
And this leads to something called the threshold effect.
Yes, a really critical concept for mitochondrial diseases. See, each cell has hundreds, maybe thousands of mitochondria, and each mitochondrion has multiple copies of mtDNA.
So it's not all or nothing.
Not at all. A mutation might only affect, say, 30% of the mtDNA copies in a cell. This mixture of mutated and normal mtDNA is called heteroplasmy.
Okay.
Disease symptoms usually only appear when the percentage of mutant mtDNA copies crosses a certain critical threshold in a particular tissue, and because mitochondria are about energy.
The high-energy tissues get hit first. Brain, muscle, heart.
Exactly. CNS, muscle, heart, liver, kidneys. Those are often the first to show problems when the energy supply dips below that threshold.
Can you give us an example?
A classic one is Leber hereditary optic neuropathy, LHON. It causes progressive vision loss in both eyes, usually starting quite suddenly in young adults, maybe 15 to 25 years old. Purely mitochondrial, purely maternal inheritance.
All right, so we've covered repeat expansion, mitochondrial DNA. What's the third non-classic pattern? Genomic imprinting. This sounds intriguing, a chromosome remembering where it came from.
It is intriguing. Genomic imprinting is an epigenetic thing. Think of it like temporary chemical tags put on the DNA during egg or sperm formation. These tags silence specific genes.
So for certain imprinted genes, you only have one working copy, either mom's or dad's.
Precisely. The other one is switched off by these epigenetic marks.
And the classic example pair here
is Prader-Willi syndrome, PWS, and Angelman syndrome, AS. They are two completely different disorders, but the genetic fault lies in the exact same neighborhood on chromosome 15.
Okay, walk us through Prader-Willi first, PWS.
In the PWS region, the genes on the copy you inherit from your mother are normally silenced or imprinted. They're switched off.
So you rely entirely on the copy from dad for those genes?
Correct. So if the paternal copy has a deletion or some other defect rendering it non-functional,
You get Prader-Willi syndrome.
you get PWS, which involves intellectual disability, very low muscle tone, or hypotonia, especially in infancy. And this characteristic, insatiable hunger, hyperphagia, leading to severe obesity if not managed. Also, typically small hands and feet.
Okay. Now flip it, Angelman syndrome, AS, same region of chromosome 15.
Right. But for the key gene involved in Angelman syndrome, which is called UBE3A, it's the copy from the father that's normally silenced, at least in the brain.
So for brain function, you rely solely on mom's copy of UBE3A.
Exactly. So if the maternal copy is deleted or defective,
You get Angelman syndrome.
you get AS, which also involves intellectual disability, but the features are very different. Microcephaly, small head, problems with balance and movement, ataxic gait. Seizures are common, and this unusual characteristic happy demeanor with frequent, often inappropriate laughter. Hence the old and rather problematic nickname, Happy Puppets.
It's just wild. The exact same missing piece of DNA causes PWS if it's lost from dad's chromosome 15 and AS if it's lost from mom's. That really hammers home the parent of origin effect.
It absolutely does. And we know a bit about why now, at least for Angelman, that UB3A gene. It codes for a protein called a ubiquitin ligase, which is involved in tagging other proteins for disposal.
Okay.
And in the brain, it seems to be critical for normal synapse development and plasticity. Losing the maternal copy messes that up.
So it highlights just how crucial gene dosage is and how epigenetics can just silence one copy completely.
Right. Even though you have the other copy, it's effectively invisible to the cell because of the imprinting.
Okay, that brings us to our fourth and final category, gonadal mosaicism. You said this one is a nightmare for genetic counseling. Why?
Because it messes with our basic assumptions about recurrence risk. Let's say a couple has a child with a severe autosomal dominant disorder, like osteogenesis imperfecta, brittle bone disease.
Yeah.
But both parents are perfectly healthy, show no signs of it.
Right. You'd usually assume it was a brand new mutation that happened spontaneously in the egg or sperm or right after fertilization.
Exactly, a de novo mutation. And you'd tell the parents the risk of it happening again in another child is very, very low, basically the background risk for anyone.
But gonadal mosaicism changes that calculation.
Completely. Gonadal mosaicism means the mutation happened after fertilization, in the very early embryo, but only in the lineage of cells destined to become the eggs or sperm, the germline.
So the rest of the parent's body cells, their somatic cells, are normal. They look and feel totally healthy.
Correct. Phenotypically normal. But a portion of their sperm or eggs carries mutation.
Oh, so they can pass it on again.
They can. And the risk isn't negligible anymore. It could be, theoretically, up to 50% for each subsequent child, just like a typical dominant disorder. Even though the parent seems unaffected, you just don't know what proportion of their germ cells carry the mutation.
That is a counseling nightmare. How often does this sort of thing happen?
It's thought to account for a noticeable percentage of cases where apparently unaffected parents have more than one child with a severe autosomal dominant condition. It's definitely something geneticists have to consider.
Okay, so bringing this all together with modern diagnostics. Things have obviously moved on from just looking at chromosomes under a microscope, right?
Oh, absolutely. Especially for these kinds of disorders. You know, for Fragile X, they used to try and spot a physical fragile site on the X chromosome using specific cell culture methods. Very tricky.
And now?
Now it's molecular. For trinucleotide repeats, we use PCR-based methods to precisely measure the number of repeats. It's quantitative. For mitochondrial disorders or suspected imprinting defects, it's often comprehensive sequencing of the mitochondrial genome or targeted analysis of the relevant regions on chromosomes like 15. Much more precise.
So that level of detail is crucial for diagnosis and management.
Mandatory, really.
Okay, so wrapping this up, what's the big picture takeaway for everyone listening? Non-classic inheritance really forces us to think way beyond those simple dominant recessive Punnett squares.
Definitely.
Yeah.
You have to ask, which parent did the gene come from?
Yeah.
How many internal repeats does it have? Is the DNA even in the nucleus or is it in the mitochondria? These factors fundamentally change the outcome.
Right.
And these mechanisms, toxic RNA and pre-mutations, the silencing effect of imprinting, the mitochondrial threshold effect, they are really reshaping how we understand, diagnose, and hopefully eventually treat a whole range of developmental and neurodegenerative diseases.
Yeah.
We're moving towards targeting these very specific molecular problems.
It's incredibly complex, but also fascinating. So let's end with a final provocative thought for you, the listener. We've talked about genomic imprinting, how certain genes effectively know if they came from mom or dad, and how that dictates major disease outcomes. So the question to mull over is, what are the broader, much more subtle impacts of these parent origin effects on just normal human variation. Things like height, maybe aspects of intelligence, maybe even personality traits, which are influenced by many genes. Could some of those genes also carry an imprint, a subtle bias based on which parent they came from that we haven't even begun to untangle yet?
A very interesting question indeed.
Something for you to think about until our next deep dive.